So now I've put a slide on that only has fluorescent stains on it, unlike this, where we can see these vital dyes already in the tissue, staining it red and staining nuclei blue, etc. These are not fluorescent dyes. These are dyes like the dyes in my red shirt. So, this is not fluorescent. Now I've got on the screen here an image of what tissue looks like when it's not fluorescent. Your skin, your tissue, your cells are basically see-through. They're basically minuscule water bags. So is this a cell? Is this a cell? Is this a cell? You'll never find a cell that beautifully round. Those are air bubbles that got trapped under the cover slip. Remember how I didn't do that great of a job last time when I was putting the cover slip on? This is what happens. But luckily our piece of tissue is right here. Well then what's this? What's this? What's this? What's that? Those are things like dust from the ceiling when you were doing the cover slipping, or little bits of fragments of skin falling off, as your skin cells always are doing all day long, or human hair, threads from your clothes. All of these are background garbage. So where's my tissue? This is the tissue that I want to see. This is axolotl tissue. You can see these black spots. Those are the little bits of pigment in the axolotl skin. So I'm at Brightfield right now. Let's go to we'll go up here and we'll switch to a filter setting that allows only light to come in that is like a green spotlight, okay? So it actually comes out as a blue-green spotlight that will give off green fluorescence if there's anything to see there. So I hit Fitzy, and we have some changing here in the filter sets. Now you can see that the light has changed. Instead of white light down here from the bottom, now I've got blue light shining from the top. Remember that our Excite Mini makes the light and shoots it through this fiber optic cable. It comes in here and is focused as a beam of light right down, covering my whole tissue section. That beam of light is bigger than my piece of tissue. So here it is, blue coming in, very energetic. What's coming out? Well, we don't see anything coming out because we're at 381 microseconds, which is not a long enough exposure time. So let's go, let's do 5 seconds of exposure time. Let the camera see all the green that is coming off. This is background, folks. This is, we let the camera look for so long, of course it thought it was seeing something. So what we really need to do is we need to find the balance of exposure time here. So what we can do quickly is put on the super fluorescence, which means that it knows that it's going to be super faint, because fluorescent isn't nearly as bright as our normal lights, and switch it to automatic and see if it will lower the time there. It's not doing it. Not doing it fast enough for my liking. Now it's down to 1.2 seconds. Why? You know why it took so long? Because I didn't wait 5.5 seconds. This is not milliseconds or microseconds, it was seconds. So now every 1 second it will update the image. So I'm going to move this stage now. Watch the screen. I'm moving the stage. And now you see it move. I'm moving the stage. And now you see it move. I moved. I moved. I moved. I moved. This is awful. Right? All you're going to get is a headache looking at things moving this slow. Well, what we actually care about is the green here. We don't want any of this green out around the water molecule, the air bubbles. We know that this is the green that we care about. So let's go ahead and grab this slider bar and manually adjust it so that the green that we can see is the green in the tissue and not the green so much out in the background. So here is some staining in the green. I'm at 4x though. So we're going to want to look at this at a higher magnification because the light is so faint that we're still getting tons of background. Let's see what the other colors look like. So if I come up here, I go from Fitzy to Tritzy. Tritzy is the green light coming down. Just look over here. Here's my green pillar of light. Okay. And that's going to excite and give us a red image. So here we can see very bright red. 713 milliseconds, 0.7 of a second is way too much. Oh, now we're starting to see individual cells. Can you see that round cell right there? Little circles of each of these individual cells. Now notice how the background is going away as I get into the range where fluorescence usually should happen. Fluorescence, you want to be in the 100 to 500 millisecond range. Whereas with visible light, the bright field, we're at 500 microseconds, not milliseconds. So that's 1,000-fold brighter in the bright field. Okay, so now we can see what our red looks like, and we can go to the DAPI setting. And, oh darn, why can't I see anything? What is that piece of garbage over there? I wanted to see my tissue section. Oh, trust me, there are nuclei there. And blue, this is using DAPI to stain the nuclei. There are nuclei there. Let's just up the exposure time. Let me see if the automatic will get it there for me now. 33. Bummer. Exposure is not getting it as high as I want it to, so I'm going to go ahead and bump it up more than that. Not that much. I got to wait. When I get to 1 second, I have to wait 1 second before I expect the computer to look at what I'm seeing. Do you see all those blue dots like a starry night? Those are all nuclei of cells. Look at all these big cells in the middle and all these skin cells around the outside. There are a lot of cells there. And what did we see in the FITSI? FITSI is the green filter set. FITSI stands for fluorescein, which was the first green dye, fluorescent green dye. Yeah, we can see a lot of those cells are there. Of course, if we go to the TRITSI, which is the red-orange filter set, we have to bring that way down. When it turns yellow here, that's overexposed. That is not what it actually looks like. So let's bring it down into the 100 range. And we have these 3 images. Now, let's try to look a little bit closer and see what's going on in these cells. In fact, I want to focus on these cells in the middle, because those were pretty interesting under the DAPI. So I'm going to jump. I know I'm in focus at 4x. I'm going to jump to 20x. I've got this relatively in the center. Yeah, not so much in focus anymore, is it, right? So you got to be careful that you don't jump too much before you lose focus. What is this? See a cell with red? It's so much red. Now that's 140 at a higher magnification is too much light. The camera can't handle it because we're so focused on a smaller area that now the light is extremely bright. What do you see here? These little hexagons. This red is in the membrane of each of these cells. I'm going to move the stage over and see if we can't. We're very close to that air bubble still. So let's move into our tissue. So now that piece of tissue was sort of sitting on top, and now I'm going to have to focus down. Ooh, wow. What do you see here? There's sort of a hole in the center with a bunch of cells radiating out from it. This is the spinal cord of a salamander. And we're looking at it right down, like looking down through a rod or through a barrel of a gun or something. Here's the center of the spinal cord where cerebrospinal fluid will flow, and here's all the different cells. In an axolotl at this stage, this region is called the ependyma. Okay? And the ependyma can produce new neurons for things like regeneration. We've got a lot of other cells out here in the connective tissue around it. So we might have bone and spinal cord nearby to the spinal cord. So we can get this red image. Let me move just a little bit more to the side so that bright region. Oh, I got another bright region over here. Those are skin cells that are so bright on the right. I'm going to try to just focus on this region so that I don't have that bright background. And now I feel like I can up it a little bit brighter, maybe too much, so I can see each individual cell. Now what I'm going to do is something I haven't done yet. I'm going to take a picture of this. So here I am on live. I'm going to go to the snapshot. Notice that it saved that image. But without changing the focal plane, I am now going to change to the DAPI, and I'll turn up the exposure time. There are all the nuclei in that exact same field of view, so I'm going to take a snapshot of that. And let's not be done. Let's go to the FITC, which is the green. And well, it looks like that's a pretty good exposure time. I'm going to up it. That might be too much. This is where science becomes art. You have to have an aesthetic sense of what will look good together. What am I trying to show in my scientific observation? So I'll snapshot it like this. The green is pretty bright. The blue is a little faint. The red is sort of medium. And now what I'm going to do is I'm going to come up here to the things that this program can do. I've already taught you how it does stitching. Now I'm going to teach you, I'm on the live. It won't do anything until the live images. I need to select the red, the blue, or the green image. And I want to overlay these to each other. So I'm going to combine color images. Here's the red only, but now I have drop-down menus. Maybe instead of just one layer, the red layer, maybe I'd like to add a blue layer to that. And maybe on top of that, I'd like to add a green layer. So this is showing me a snapshot of what is going on as I lay these over each other. Over here, the intensity, I can, let's say that I feel like my blue is actually a little too bright. I can pull back the blue by post-processing here. Maybe I want to, I see some blue, I see some purple, I see some red, but only right over here do I really see some green. Let's say I want to bring out some more green. Then I can up this. Notice how the green background is the first thing to come up. I think if you look down at this image, you can see all that green background. I don't think I want to bring up the green. I think maybe a better way to make a contrast is to lower the red. Now can you see where there is more green here and more red here? Can you see this cell with green around the outside? Here's a good one. There's green around the nucleus with blue in the center. So I'm going to say, okay, that looks okay. Let's see what the final image looks like. I want to make sure to merge the layers so that it will save it as a fourth image, as an image of all three together with all three colors. I'll apply that, and here's image 01. Just save that image. So I'll close that now, and let's get a good view of what our final picture looks like. So I've got green in a few cells. This cell has more green. There's green more around here. I've got red in between some cells here in this area. I've got blue in the nuclei. Depending on which proteins I'm looking for, I can see cells that have different properties. This has red and blue and green. This has just red and blue. I don't see green. This has just green. I don't see blue or red. So when looking for different proteins, we can use these overlaid images to find where our proteins are in relation to other images. This is called an overlay, or a combine of the color images. So we have adjusted the image in a post-processing way, and then we save all these images, the combined image and the original images, so that if we ever go back and say, oh, there's another cell here that I didn't notice, I need to bring that cell out more, we can play with the adjustment of how bright is the green versus the red versus the blue, and be able to document that something is going on in our tissue. So here is some regenerating salamander spinal cord tissue. And what we've done is we've used epifluorescence to find that tissue. So now I've taken our slides that we actually processed, and I put one of them on here. And here's what it looks like at 10x, at bright field. This grayish blue on the individual cells, that's the autofluorescence quencher. which it looks like didn't fully wash off. So I'm expecting that we will not see very bright DAPI in these regions because there's too much autofluorescence quencher. This is something that takes a lot of practice. The other fact about this tissue is you can see that it's sort of folded over here, all these little lines across it. I wish I'd used the expensive slides so that the tissue would have stuck better. We also have this brown region here. This brown region is probably a sign of drying out of the tissue during maybe the overnight 4-degree incubation with the primary antibody, where things like the blocking solution dried onto the surface of our section. So this area out here, right, we might be able to see something interesting. Let's go ahead and switch. In other words, I'm saying there's a lot that's not optimal in this section. Let's see if we can still see anything. Let's go ahead and go from bright field onto, let's do Tritzy first, which is our red color here. Well, I don't see anything at microseconds, but we're not supposed to. Fluorescence needs to be in the millisecond range. And so we'll bring it up to, oh wow. Look at how well our wheat germ agglutinin stain bound to our cells. Can you see this network of cells? Every single cell has been surrounded by a round membrane. What is this? This tree branch, this leg, this stick figure? Those are blood vessels. And those are the cell membranes of endothelial cells. What's this huge thing right here, this log sitting in our forest of cells? That's a muscle cell. Muscle cells are long and fibrous. So here you can see the membrane of one muscle cell next to the membrane of another muscle cell. Wow, that wheat germ agglutinin worked fantastic. It bound to every one of our cells and showed us exactly where their cell membranes are. So now what do we expect to find on the inside? Well, we'd expect there to be a nucleus in there. So let's go to DAPI and see. I bet we can't see with all that autofluorescence quencher. Oh, I see a little bit of blue over here and a little here. We're going to have to pump this up. Let's go into the second range rather than the millisecond range to see where is their DAPI. Aha! So I saw a lot of autofluorescence quencher in this region, but out here, each of those blue spots is a nucleus of a single cell. And you can see them in here even though the quencher is making them shine not as brightly as usual. We were trying to block out the background of like red blood cells, etc. It appears we've done that, but for whatever reason, either the concentration of the blocker of the autofluorescence quencher was too high, or we left it on too long before we rinsed it off with PBS. The blue signal is too faint here. We can see it, we could get a picture of it, but it's fainter than we would like. Finally, we want to look and see, in the FITC channel, is our green antibody present? Well, there's definitely some green there, but notice that all of these individual cells, this whole area is equally green in the background. That is background signal. We want to go back down to the millisecond range. Why do we see a starry night of green stars? This is what unbound antibody looks like. This is our secondary antibody, which we put in at a very high concentration, that just found some sticky spot to stick on our slides. So this is not, if you saw the pattern of a cell or the cytoplasm or its nucleus or its cell membrane, you would know that this was real staining. But this very punctate globs of antibody are just stuck into random spots. This is not real signal. This is the background of the fact that we're using a molecule, an antibody, that is naturally sticky, and it's stuck to parts of our slide. So overall, this demonstration of of the slides that I made, I used the cheapest slides. I rushed through the procedure so that I could show it to you in a reasonable amount of time. We would need to really clean up our procedure in order to get a better result than what we see here, which I have done many times. So now we're going to go and use the confocal microscope to analyze a tissue section that has very nice staining of our different antibodies.